Okay, thank you very much for inviting me to come here and give a presentation. And the talk this afternoon will be about what we call ancient DNA. It means the retrieval and analysis of the genetic information from ancient materials and fossil remains. And it's actually a quite young research field. It's only about 20 years old, and it operates, you can say, on the interface between molecular biology, paleontology, geology, and archaeology. So it's truly multidisciplinary in nature. So I'll start this talk by giving you a very brief introduction, historical introduction to the field, and then I will continue with some of the research projects that my group has been and is involved in. Okay, so it basically all started back in 1984, where a guy called Alan Wilson from Berkeley University showed it was possible to obtain DNA from the skin of the quagga. This is a quagga. It's a zebra-like animal that lived and died out in South Africa in the end of the 1800s. And then the year after, a young Swedish student at this time, his name is Svante Pääbo, who is now one of the biggest names within human evolution. He claimed in Nature, which is this prestigious journal, that he could in fact obtain DNA from the skin of an Egyptian mummy several thousand years old, okay? And then in the end of the 80s, 1980s, beginning of the 1990s, we saw a technical revolution, which is called the polymerase chain reaction, which basically for the first time made it possible to retrieve, you can say, single surviving DNA molecules, okay? And this really revolutionized molecular biology as such, but also had a major impact, of course, on ancient DNA research. And in the beginning of the 1990s, you could read in high-profile journals like Nature and Science, well, it's possible to obtain DNA from amber entombed insect, dinosaur bones, magnolia leaves, all millions of years old. So there seemed no limitation to what you could actually obtain genetic information from, right? However, after a few years, it actually turned out that this DNA sequence from the Egyptian mummy probably came from Svante Pääbo himself. Okay? And after a few more years, well, then it turned out that all we call geological ancient DNA, it means DNA that is more than 1 million years of age, was in fact the result of either human contamination, it wasn't Svante Pääbo in all cases, I must say, and in one case probably somebody eating a chicken sandwich within the laboratory, right? So you can say quite early on it became evident that it's very easy to obtain what we call false positive results due to contamination with contemporary DNA. So why is this the case? Well, the DNA molecule, which actually creates, I should probably mention that, which actually makes us who we are, right? Is actually a fairly unstable molecule, so it will decay with time if it's not repaired. And this is due to various processes, bacteria eating the DNA in the sediments, nucleases chopping up the DNA, but even if it survives those processes, it will still decay due to spontaneous chemical reactions called, for example, hydrolysis and oxidation, which will take place spontaneously in the presence of water or oxygen. And in fact, even when I'm talking right now, the DNA in my cells will constantly be damaged due to these degradation processes, and I will probably die before I reached home if it wasn't due to an extensive repair system that goes in and repair all these damages, okay? However, when an organism dies, the repair system stops functioning while these spontaneous degradation processes, they continue. So the speed by which this degradation takes place is highly dependent, you can say, on the exact environmental conditions in which the DNA is preserved. And this really, embarrassingly enough, makes it impossible for us at the moment to say precisely how long time can DNA survive under various environmental settings. Many people have tried, including myself, with all kinds of models, and every time we find out, well, we were wrong, you know. However, what we do know today is that in general, some areas are better, some environments are better of long-term preservation of DNA than others. For example, it's very good to be under frozen conditions because all the chemical reaction goes on very slowly. It's also good to be under cold conditions. quite surprisingly, it's actually not too bad to be under hot, but very dry conditions. Their degradation also seems to be slowed down. What is really bad is hot and humid conditions, like in the tropics, for example. So there we are talking about maybe a DNA survival of a few hundred to a few thousand years, while under frozen conditions, well, the record, which I got, is around 500,000 years. But I wouldn't exclude the possibility. We are working, for example, at the moment on ice samples from Antarctica, where the temperature is -50 and has stayed that low for hundreds of thousands, maybe millions of years. Maybe their DNA can survive, maybe for millions of years. Who knows? We will find out soon. Okay, so maybe we can't obtain DNA from amber and tumbit insect or magnolia leaves, but what kind of material can we in fact obtain ancient DNA from? Well, we can obtain it from bones and teeth of animals and humans. We can also obtain ancient DNA from soft tissue of animal, humans, and plants, and that includes things like hair. And it's not, you don't need to have the hair root, which is the living part of the hair. It's actually enough to have the hair shaft. It's in fact an excellent source of ancient DNA. I will get back to that later. It also contains things like ancient feces, ancient shit, which is what we call coprolites in scientific terms. And then it in fact also includes things like ancient sediments and ancient ice, where it has been proven possible to obtain DNA from animals and plants, also higher animals and plants like mammals and birds, even in the absence of any macrofossil evidence in the form of bones or hair, etc. I'll get back to that also later in the talk. So how do we do it? Well, honestly speaking, it's not rocket science. It's actually fairly simple, right? I mean, you basically, you take a piece of bone, you grind it up into bone powder, and now I didn't have any picture of bone powder, so this is cocaine on a glass plate, but it's pretty much, it looks pretty similar. Well, then you basically add some reagents, some chemicals, lysing all the cells, releasing the DNA, you know, the genetic information, so we can get removed every other cellular parts like cell walls, proteins, etc. And then today we are using, you know, a new technological revolution. I'll get back a little bit more into this later in the talk, called these second generation sequencing machines, which allow us to sequence, you know, get the genetic code out, so to speak. I mean, in massive amounts over a very, very short time. It has been, you can say, what the PCR, what the polymerase chain reaction was in the late 80s. This is what these second generation sequencing machines are for modern molecular biology today. But we can use them in different ways, and I won't go into much details, but you can either do what is called a shotgun sequencing, what we're using when we are, for example, doing a genome. There you're sequencing randomly what you have in your DNA extract, or you can actually go in various ways and target specific regions of the DNA you then afterwards sequence. And this is, you can say, you typically end up with some kind of picture like this. This is what you see also in CSI, right? It's actually, you can say this is the order of the 4 different bases which decide that you are you and me, I am myself, right? And this is a chimp, etc. And it's actually only the order of 4 different bases together. And so what makes it problematic is that it's basically some old shit that we're working with, right? So it's highly degraded, and that creates a number of problems. There's inhibition of the polymerase chain reaction due to degradation products. There's DNA damages, and some of these DNA damages can in fact result in errors in the DNA sequence you get out, so you actually get the wrong code out, right? And then, of course, the biggest issue is contamination with modern DNA, right? So you get these false positive results. And I won't get into major details, but let me just put it this way. We're completely paranoid with contamination. So, for example, we are using these full body suits, face masks, special labs with positive air pressure and nightly UV radiation to erase all the DNA, right? And these labs are separated physically from any other modern biological lab, you know, in order to reduce any contamination, you know, provided by the researcher himself.